Hello, welcome to our second video for today. So I'm going to start off by asking you to do a Kahoot. If you've never used Kahoot before, Kahoot is a tool where you could take a quiz. It's all multiple choice. I will not be grading you on this, but I ask that everyone do complete it. Just have to click through this, and I believe you need to make an account. I will be able to see what everyone answered, but again, I will not be grading you on this per se, although maybe the person who gets the best score will get to be biology champion for a day. Never know. I don't know. And that is just a review for things that we've already gone through in class. So I try and get people to keep going through the stuff that we've already covered so that you don't forget anything, nothing gets left behind, because we do move at a fast pace through this class, as you guys already know. So right now we're going to be talking about DNA replication. And so overall, the mode of replication for DNA, which we know is correct, is semi-conservative. And this just means that in every resulting strand or double helix of DNA, you have one original strand. So here we see the orange. In the resulting helix, we have one orange, and we have one new strand of DNA. So that's one is red. And so I'd like to pause for one moment and just talk about the vocabulary for a moment. So we have two strands of DNA. So many times I see students writing strains. So strains are genetic variants of some type of organism. Strands, and I apologize if I'm not being very clear with my enunciation. Strands are one half of a DNA helix, which consists of half of the base pairs in a double helix, as well as one DNA backbone. And we, as I said, we know that semi-conservative replication occurs, whereas the other possibilities that were theorized prior to us knowing this were dispersive, which meant that DNA replication would begin and end multiple times on both strands, so you'd get a mix of new and old DNA, but you would have a mix on every strand. And once we go through DNA replication, you're going to see that if this were true, it would just be a disaster. It would be really, really difficult to study. So thank goodness that this is not the case. And then the other option was conservative, where you would take apart the two strands of a double helix and then copy both of the strands and then separate the new and the old and put the two old back together and put the two new together. So that also adds an unnecessary step. So this is not true either. So we're going to talk about a number of different proteins that contribute to DNA replication. So some of these proteins are going to work similar to machines. So some of them are enzymes that just perform a function or catalyze a reaction over and over again. Other proteins that we're going to talk about are more for stabilization. They don't perform a very important catalytic process, but they are still crucial for the success of replication. And other proteins will not actually interact with DNA, but they interact with proteins that interact with DNA. So they're one step removed. So just as a review again, this is a depiction of DNA. And this is a great image because it shows you all of the chemical groups of DNA going in both directions. And so we see here that the double helix of DNA has each strand is reversed with respect to the direction of the other strand. What does this mean? All right, so it means that on the strand on the left here, we have the 5' end at the top. What is the 5' end? Review here. Okay, so this is a phosphate group. The 5' end has the phosphate group. That is, you can't add any new nucleotides to that end. That's just a dead end. It's the starting point for the DNA. Down here, we have the 3' end. The 3' end ends up with a hydroxyl. And you can add to this end. So take a good look. I think this is an excellent depiction, again, because here we see the phosphate group at the top, and we see the OH group down here at the bottom. Of course, this nucleotide, the last nucleotide to be added, is also going to have a 5' end, but that 5' end is going to bind to the OH here. So this is showing the process of adding a nucleotide, and we're going to end up only with the OH being available. And that's the 3' end. This is the end where you could add new nucleotides. Now the other strand, which here is called the template strand, goes in the opposite direction. So we have the 5' end here, the phosphate group, and then at the top we have the 3' end, the OH group. And so I kind of cut off the image here, or I guess they did. And this is an oxygen, but you could imply that another nucleotide is attached here, or that this could end in an OH. Either way, this is the 3' end. You could add to this end of the DNA. You can't add to this end of the DNA. And the reason why this is just the structure of the molecules themselves. The two strands bind through their nitrogenous bases, and these are our hydrogen bonds. And when you bind DNA strands together like that, they must be running anti-parallel or reverse to one another. So again, this is called an anti-parallel structure of the strands of DNA, which means that you have the 5' at the top of one strand, the 3' at the top of the other strand. Okay, so we're going to meet DNA polymerase now. Now, polymerases are molecules, they're enzymes that make polymers. So I'll say that again. A polymerase is an enzyme that makes polymers. What is an enzyme? What type of molecule is an enzyme? What type of monomers does an enzyme have? So I hope that you know this, but to answer my own question, an enzyme is a protein, specifically a subset of proteins that has some sort of catalytic ability. And since an enzyme is a protein, that means that the monomers are amino acids. Now in this case, this enzyme can add monomers of nucleotides, nucleic acids, onto a DNA strand. Again, it's a little bit confusing when you try and describe it in that way, so you can gradually build up to that. Kind of think of it in your own words in your head, and then practice explaining it using the scientific terminology. So again, this enzyme is called DNA polymerase. We're going to see different types of polymerase throughout the course, and most of them are called blank polymerase. So this one is DNA polymerase. And this is the most important enzyme of DNA replication because it adds the nucleotides. You can't make DNA unless you're adding nucleotides. And how does it add the nucleotides? Well, it's going to read, quote unquote, the template strand of DNA, and then it's going to add to the nascent strand or the new strand of DNA. So the first step is for, as we said, DNA polymerase to read the template strand of DNA. And so this is not actually the DNA polymerase reading anything. What happens is the DNA polymerase kind of pulls in whatever nucleotide is around, and it positions it so that it can hydrogen bond with the nucleotide that is the next one on the template strand. So this is a randomly chosen incoming nucleotide here, and the DNA polymerase positions that so that it's going to be hydrogen bonding with this here. And so not any nucleotide is going to correctly hydrogen bond to whatever nucleotide is on the template strand. It's only the nucleotide which is going to, that follows the base pairing rules, right? So if this is a T here, only an A is going to appropriately hydrogen bond with it. A G or a C or even another T will not form the correct hydrogen bonds. And if the correct hydrogen bonds are not made, then that nucleotide is not going to be stabilized. And so it's going to be knocked out of position. If it is stabilized, then that means, or it should mean, that that's the correct nucleotide to match up with the template strand. And then we're going to actually kind of cement that nucleotide into place. So you'll notice that the nucleotide that is incoming has 3 phosphates. So this is called a nucleoside triphosphate. When we're talking about the backbone of DNA, we only have 1 phosphate in between each of the nucleotides, right? Which kind of leads us to the conclusion that we're going to get rid of 2 of these 3 phosphates. And that is actually true. So 2 of the phosphates are going to be removed, and these are removed by the DNA polymerase. And this actually results in a release of energy, which allows the DNA polymerase to then form a bond between this incoming nucleotide and the growing strand. So the bond is going to be a phosphodiester bond, which is the type of bond that we see between these nucleotides. And so we see here, we have a phosphate group that is bound to the hydroxyl group. So in each case here, we see the phosphodiester bond on both sides of the chain of DNA. And so the DNA polymerase is what actually forms those phosphodiester bonds, or causes the formation of those phosphodiester bonds. And again, it only does this if the nucleotide that has been put into position actually fits there correctly. If it doesn't fit, then that's going to be spit out and a new nucleotide is going to come in. And so this is just a depiction of that. So we release 2 of the phosphate groups, and then they actually are separated, and that all results in a release of energy. And then the DNA polymerase will move on and quote unquote read the next nucleotide on the template strand. Again, it's not actually reading anything. There's a hit or miss kind of method. Incoming nucleotide comes, does it hydrogen bond correctly? Yes, it stays, bond is formed. No, it gets kicked out, next nucleotide comes in. We try another match for that same template strand nucleotide. And here we could see a really good depiction of how we read in the opposite direction of synthesis. So we are reading the bottom strand and we are synthesizing the top strand. So the 5' phosphate is going to come in here to notch into this 3' end, and then we're going to have a new 3' end. So we're going from 5 to 3, 5 to 3. This opposite strand, we're using it in the opposite direction. So we started out with 3' and we're going towards the 5' end. So again, keep in mind what end you're moving towards, or which one is being made. So in this case, we're always making a new 3' end. On the bottom here, we're moving towards the 5' end. So as it says here, we synthesize 5' to 3', we read 3' to 5'. So we would say that DNA polymerase has 3' to 5' reading capacity and 5' to 3' synthesis. Okay, and so I want to point out that, again, going back to here, these hydrogen bonds form spontaneously. they stabilize the molecule. You don't need energy, or you don't need the energy of an ATP to form these bonds. However, the phosphodiester bonds do need energy to be formed. These are covalent bonds. The hydrogen bonds are not covalent. And so this is why the catalytic activity of DNA polymerase is to synthesize these phosphodiester bonds, not to synthesize hydrogen bonds. Okay. So we kind of already went through this, but this will be good for your studying. So we just went through a really simplified model of DNA replication. We are only talking about the replication of one strand of DNA as the template. But we didn't show a couple of things. So we didn't show how replication starts out. We kind of showed the process in the middle. We didn't show, as I said, both strands of DNA being copied at the same time. And we didn't talk about copying the ends of the chromosomes. So replication actually begins at specific places in the genome. And these are called origins of replication. And these origins of replication are regions of DNA that have specific types of nucleotides. So we said, I think it was last week, that GC pairs have 3 hydrogen bonds, whereas AT pairs have 2. And we see that again. The CG pair here has 3 hydrogen bonds, the AT pair has 2. And that always occurs whenever you have CG and AT. And so these origins of replication are high in AT pairs, and that makes them easier to pull apart. And so when we pull them apart, there are these proteins called single-stranded binding proteins that help to stabilize the helix from coming back together, because again, those hydrogen bonds can spontaneously reform. And it does take some energy to pull apart these strands of DNA. But remember, we're not breaking any covalent bonds when we're melting apart or peeling apart the 2 strands of the double helix. We're just breaking hydrogen bonds. So this is done by initiator proteins. So these guys, these purple guys, are initiator proteins. They're what actually open up the replication of origin. And then, as I said, subsequently, we have these single-stranded binding proteins, which stabilize the DNA and prevent it from spontaneously reforming those hydrogen bonds. Then, the enzyme that a lot of people are familiar with is helicase. So helicase does not do the initial opening at the replication origin, but helicase does do the subsequent breaking of hydrogen bonds. So again, remember, helicase does not break covalent bonds. It breaks hydrogen bonds, which are very weak, and it breaks them one at a time, and that's how it gets around this problem of the DNA strands actually being held together quite tightly due to the number of hydrogen bonds. So helicase moves in one direction, and we actually end up having multiple helicase molecules. So as helicase works on the DNA, peeling apart those 2 strands, if you think about it, if you have a twist tie or 2 shoelaces or something, or a lanyard, and you twirl it up, and if you have a friend that's going to peel it apart in the middle, then the 2 ends are going to get more and more tightly wound. So if you do it tightly enough, what's going to happen? Well, eventually something's going to break one of the strands. We don't want our DNA strands to break, because that would be bad for our genetic material. So there is another enzyme called topoisomerase. And topoisomerase actually intentionally breaks the DNA. So it breaks the phosphodiester bonds in one place, or sorry, the phosphodiester bond in one area of the DNA. So it's just separating one single nucleotide on one strand from the next nucleotide, and then it allows the DNA to unwind in a controlled manner so that then the tension is released and we get rid of all this kind of tangling that could result in, again, the breakage of DNA. And then we actually do reform this phosphodiester bond. All right. So we talked about the start of replication, and now let's talk about copying both strands of DNA in opposite directions. So this is where it can get pretty tricky. And here, so we have one end of the replication fork. So actually I would like to go back to where we talked about the initiator proteins. So we see here what is forming is a replication bubble. So many of you probably came in with the knowledge of the term replication fork. So we have a replication fork on the left. We also have a replication fork on the right because we are opening two strands of DNA in the middle, and we don't have either at the end of the DNA. So in reality, we're going to have two replication forks from every origin of replication. So we're going to have one that looks like this and the other one which looks like this, one going in each direction. All right, had to take a quick cat break. All right, so we have one going in each direction, right? And we see here we have the darker blue, which represents the template of DNA. And so this is the original DNA where we had the origin of replication and which was pulled apart. This is the other strand of that original DNA. Again, this is another template strand of DNA. Then in the middle here with the lighter blue or gray, we have the new strands or the nascent strands of DNA. And we are going to have one DNA polymerase for each of these. So we have one DNA polymerase, which is moving along this bottom strand. And this one we see is moving 3' to 5'. That's how it reads. And then we see that it's oriented correctly so that it's adding on consistently to the 3' end as it moves towards the replication fork. As it's moving in this direction, the helicase is also moving ahead of it, separating more and more of the DNA. So that's great. This is the leading strand, or what I like to call the easy strand. Then over here we have the lagging strand, or I think that it's referred to as the pain in the butt strand by one of the videos I'm going to link because it's a little bit more complicated. So this strand is called the lagging strand because as the helicase moves in this direction, it's separating the DNA, but the DNA polymerase on this strand is moving in this direction, right? So it's not able to quickly utilize the newly pulled apart DNA. So it's reading in the correct direction. It's reading 3' to 5', and then it is synthesizing 5' to 3'. But as new DNA is revealed to be used as this template strand over here by helicase, then this DNA polymerase will come in at, we'll say here, if this has been pulled apart, and then it could move up to here. Now it's going to hit a piece of DNA that has already been replicated, and it will have to start over here where the helicase has revealed or pulled apart even more DNA. And so this is why this is called the lagging strand. It's because the DNA polymerase has to keep on going back and forth as the helicase forms, or not forms, pulls apart more and more DNA. And these shorter lengths of DNA are called Okazaki fragments after the couple that that discovered them, the relationship goals, discovering new things occurring in molecular biology and naming them after you. It's awesome. So I have these two videos, and this one is, I believe, the crash course biology video on the bottom, which is great. I can't remember what happens before this point in the video, but I did tag that one specific place in the video. And then this one here really shows the dynamics of what's going on at both replication forks. Okay, so I'm just trying to get this video here to show you. Because the original one that I've always used in class is no longer available. So what we have here is semi-discontinuous DNA replication. And I encourage you to watch this on your own a couple of times because I think it's really helpful. And so here in the gray, we have the template DNA. Here in the orange, we have the helicase. We see the replication fork. And then we have the leading strand synthesis and then the lagging strand synthesis. And so this is opposite of what we saw on the slide, which was that the lagging strand was on the top and then the leading strand was on the bottom. That is because we reversed the polarity of the DNA in this image. Not really a big deal. And as the helicase opens up more DNA, we're going to see that we have room for the leading strand to be synthesized further by this DNA polymerase, this little red ball. Enzymes are actually more complicated than that, but you know, it's whatever. But then the lagging strand is going to have DNA polymerase come in closer to the replication fork and then move away from the replication fork so that it is moving in the 5' to 3' direction. And we see that here. Okay, and so it's adding on nucleotides. And I do want to point out that initially it showed the primase coming in and laying down an RNA primer, which I didn't mention previously, but the very start of every single new strand of DNA is going to actually be started by an RNA primer because they could start from scratch instead of adding onto a hydroxyl from an already present nucleotide. So we had primase come in, then we had DNA polymerase come in, and then that whole process is going to repeat over and over again as this helicase moves in this direction and unwinds more DNA. And the leading strand is just going to continue growing in this direction. So the leading strand is actually a lot easier to describe than the lagging strand. And the lagging strand, again, those pieces are also referred to as Okazaki fragments. Okay, so I'm going to pause this video for a moment. And again, this link I just changed to link to that video, so you could watch that here. And then the other one below, I believe, is Crash Course Biology. So as I mentioned, primase is another enzyme, and it is the only enzyme for our purposes for our class that can begin a new strand of nucleotides. So the DNA strand here that is shown is the template. Then primase can come in and set down complementary nucleotides, and it continues that short chain of nucleotides, which is actually RNA, not DNA, and it makes maybe 10 or so nucleotides in a row, and then the primase will dissociate. Now, the DNA polymerase will then come and add onto this short primer that primase has constructed. So this is called a primer, this short sequence of RNA nucleotides. Primase moves in the same direction as DNA polymerase, as we saw in that video. Synthesis is 5' to 3', reading is 3' to 5'. So in other words, primase always moves further from the 3' and closer to the 5' end of the template. So eventually, these RNA primers are going to have to be removed because you can't keep these RNA primers in your DNA. And so again, I'm just going to quickly show the video that we saw a moment ago. So on the leading strand, we have a single primer. And then on the lagging strand, we have many RNA primers in between each of these Okazaki fragments. So specifically the RNA pieces of these strands need to be removed because you can't have RNA integrated into your DNA permanently. So what we see here is the removal of these RNA primers. So here the blue is the primer, and we see that it remains after DNA polymerase finishes a DNA fragment, and the RNA primer is going to be removed. Each nucleotide will be removed. It's going to be replaced by DNA, and this is actually done by a DNA polymerase, but not the same DNA polymerase which does most of the work at the replication fork. And then we still end up having a break in the phosphodiester bond because remember, the DNA polymerase is moving 5' to 3', and it can't make that last connection there, that last bond that would connect the two different fragments of DNA. So that's actually done by this other enzyme called DNA ligase. And to ligate something is to join something, so it makes sense that this is called a ligase. All right, so moving on, we come to our final and perhaps most complicated problem with DNA replication, at least for eukaryotes. So this problem doesn't occur in prokaryotes, bacteria that have circular genomes or chromosomes. And this is copying the ends of the chromosomes. So this is very reminiscent of the last image that we saw. So this image where we see the RNA primers initially, then they're removed and we have this gap. But in this image, we're actually at the end of the chromosome, so the end of the DNA molecule. So we could get rid of this RNA primer. We could get rid of this RNA primer, and both of them could be ligated then. But this one is going to leave a gap. And so the gap we see here. So neither DNA polymerase can come and add in this direction because it needs an RNA primer, but there's really not enough of a place for an RNA primer to be put down and then for DNA polymerase to add to it. And even if we did that, then we'd have to remove the RNA again. And so we'd always have a gap remaining at the end of the lagging strand if we use the method that we just talked about. So this is where the enzyme called telomerase comes in. So telomerase is actually going to make sure that we don't lose any genetic information at the end of this strand because it's actually going to extend this strand, the template strand, and then DNA polymerase is going to use that template strand to fill in this missing piece here. So how does it extend the lagging strand? Because it can't use any template for that. Well, telomerase actually carries around a little piece of template. This is actually RNA, and it carries this RNA which has just a repeat sequence. And so many of you have probably heard about telomeres and telomerase research and how this is, the key to living forever or whatever. Not necessarily true, although it is a concern because at the ends of DNA, which is called the telomere, we can lose DNA, and we do lose DNA as the cell or as I guess a genome goes through many rounds of copying as it goes through division and mitosis over the course of a lifetime. But this is a method that is used to prevent loss of DNA before meiosis, for example. So we don't always have these telomerase enzymes active in our body, so that's part of what the research is about. But we won't go into that too much today. So the ends of the DNA, all of your chromosomes have these repeat sequences. So there are no genes here. There is just a repeat sequence. And evolution has made it be this way, or it has occurred this way through evolution. So we don't end up losing any genetic information when we lose the ends of these chromosomes, at least not initially. And so that this template can be used to kind of extend or secure these ends of the DNA. So telomerase comes in, and again, it's extending the template strand. So it extends the template strand by matching nucleotides to its own little template here that it carries around, which is kind of crazy. I feel like it just doesn't obey any of the rules. It carries around its own template, says, I'm not going to follow a template strand. I'm going to use my own template and just add nucleotides. And then it goes and does it. And then we end up with an extended template strand with just these repeat sequences. And so this is just a couple of nucleotides where maybe we have ATG, ATG, ATG. That's what I mean by a repeat sequence. And then we can have DNA polymerase come in and do its job as normal, and it's going to synthesize 5' to 3', 5' to 3' and read 3' to 5' as it reads this template strand on the top. And so then we're just using our normal process that we've used previously. Yes, we do end up having some section of the template strand which does not have a complementary nucleotide strand. But that's okay because this is just repeated nucleotides and it was just made through the use of telomerase. It doesn't contain any important genetic information. So essentially, our lagging strand should be the same length as the original template strand, and the template strand actually ends up being longer than it originally was. And that's all shown here. So I do draw this out and diagram it in this YouTube video here, which you can watch. And then, so here we have the whole crew of DNA replication enzymes and proteins. You can see them all working together. We have the helicase, the proteins that initially opened up the origin of replication have already come and gone. We have these single-stranded DNA binding proteins, which stabilize the single-stranded DNA. We have the primase, which I guess is not shown here, but we see evidence of it because we see this RNA primer. We see the DNA polymerase, the other DNA polymerase here. Also pointed out is the sliding clamp, so I think that's in your text. It's just another protein that helps the DNA polymerase to hold on to the DNA and stay bound to it. And here's the primase here. We also don't see the telomerase in this picture, and we don't see topoisomerase, both of which would be closer to the end of the DNA. Okay, so that's the whole kind of mechanical molecular process of DNA replication. And I just want to talk about one or two more things to connect this with what we talked about in mitosis and the beginning of mitosis. So DNA replication needs to be regulated, right? Because we don't want the DNA to be copied at times when the cell is not going to go through mitosis, or perhaps when there are pathogens in the cell. We don't want to be wasting resources on copying the DNA when we don't need to, or if there are mutagens in the cell or around the cell, we definitely don't want to be copying the DNA. So this process is very tightly regulated, just like we saw that the stages of mitosis were regulated and the stages of the cell cycle. And so some of the molecules that participate in this regulation are actually DNA helicase, CDC6, and the ORC. And so the ORC is a protein that binds to the origin of replication, and stands for origin recognition complex. And then CDC6 is a protein that binds to that. So they block helicase from binding to the DNA. And you're going to see this multiple times in the when we talk about gene regulation and transcription and translation. The way that we can prevent certain processes is by having another protein bind to the target molecule, in this case DNA, and specifically the origin of replication, and it's binding and it's just blocking, okay? And so what happens here is that we need to remove the ORC and CDC6 in order for DNA helicase to be able to access that origin of replication. And one of the proteins which can do this is the SCDK, when it's active, complete with its cyclin, S-cyclin. And so this SCDK cyclin complex, again, is a kinase complex. It adds phosphate groups. It can add a phosphate group to this ORC complex after the CDC has been removed, and it also adds a phosphate to the helicase, activating both of these so that then the origin of replication is peeled apart so that the DNA strands are accessible, and then the helicase continues to separate the strands of DNA. And of course, again, this only occurs when the appropriate signals are present and we have the SCDK cyclin complex. So another mechanism of regulation for DNA, the replication of DNA, is the P53 and and the P21 complex, or these two proteins. So the presence of P53 in the cell is normal, but it is degraded if it's not needed. If P53 is needed, then it's going to be activated. It is activated only when there's DNA damage. So there's some other molecule that is sensing this DNA damage, and it's then going to phosphorylate P53 to make it active. P53 then goes and binds to DNA, and it causes the expression of this gene for P21. And sorry, I just needed to remove that X. And P21 is a protein that actually inhibits the G1 SCDK. So it just kind of binds to it. Again, we talked about proteins kind of blocking or inhibiting by binding. So P21 binds to GSCDK and just prevents it from phosphorylating anything else. And this is reversible. So this P21 protein can be removed if the cell can recover, if the DNA can be fixed. If the DNA can be fixed, then we're going to stop phosphorylating additional P53 proteins, and then we're going to degrade them, and the concentration of P53 is going to go down, okay? If it goes down, then we're also going to get rid of P21, and then the G1 SCDK complex will be active again. However, if we do not fix the DNA, and then we're going to just keep on phosphorylating more and more P53, it's going to accumulate in the cell, and there's some crucial concentration where if there's enough of these proteins present, then the cell is going to enter apoptosis. And so these genes are really important in the prevention of cancer because it stops cells with damaged DNA from replicating, either through fixing the DNA or through causing them to enter apoptosis. All right, so please let me know if you have any questions. I will be checking my email this afternoon. And remember, again, we will be having a quiz tomorrow when we meet in real time at 1:30. Thanks.